Hello there, and welcome to the Sleepy Library. Tonight, we are entering the quiet wonder of the human brain, where billions of tiny messengers work in harmony to create every thought, every memory, and every dream you have ever known. The brain is not just an organ, it is the birthplace of consciousness itself, a 3-pound universe folded inside your skull. Within its wrinkled surface lie hidden worlds more intricate than any machine, more beautiful than any architecture, more mysterious than the deepest ocean. This is a journey through the silent halls where your self resides, the place where you meet the world, store what you love, and become who you are. If you enjoy these gentle explorations of science, I invite you to like, subscribe, or share a thought below. It helps others find rest here, one sleepy mind at a time. But for now, settle into your pillow. Let your shoulders drop. Let the day fall away. Let us begin. The human brain contains approximately 86 billion neurons. These tiny cells are the foundation of everything you think, feel, and remember. 86 billion is a number almost impossible to grasp. If you try to count them one by one, spending just 1 second on each, it would take you nearly 3,000 years to reach the end. Yet all of these neurons fit inside the soft folds of your brain, each one connected to thousands of others in a web of communication so vast it makes the internet look simple. Neurons are not static. They are alive, constantly reaching out to their neighbors, sending signals that create your thoughts, guide your movements, and store your memories in patterns of electricity and chemistry that shift like clouds in a summer sky. Each neuron connects to roughly 7,000 other neurons through structures called synapses. A synapse is where two neurons meet but never quite touch. There is a tiny gap between them, a space smaller than you can imagine, measuring less than 50 nanometers across. This gap is where magic happens. When an electrical signal races down a neuron, it releases chemical messengers called neurotransmitters into this microscopic space. These molecules float across the gap and land on the neighboring neuron, passing along the message. It is like whispering across a canyon, except the whisper is chemical, and the canyon is almost impossibly small. With 86 billion neurons, each making roughly 7,000 connections, your brain contains approximately 600 trillion synapses. That is more synapses in your skull than there are stars in our galaxy. Each one is a tiny junction where information flows, where decisions are made, where your reality is constructed moment by moment. In a single cubic millimeter of your brain, a space no larger than a grain of sand, there are nearly 150 million synapses. Scientists have mapped this tiny fragment of tissue using powerful microscopes and found it contains enough connections to fill thousands of terabytes of data. Almost every neuron in that space forms just one synapse with each of its neighbors, keeping the network organized and efficient. But some pairs of neurons are different. A small fraction form multiple connections to the same target, creating super strong pathways. In at least one case, more than 50 synapses connected a single pair of cells, suggesting that certain messages are so important they need extra emphasis, like underlining a sentence 50 times. These strong connections are rare, but more common than chance would predict, hinting that your brain intentionally builds highways for the information that matters most. Your brain's neurons are not randomly scattered. They are organized into layers and regions, each with a specific purpose. The outer surface of your brain is called the cerebral cortex, and it is where most of your conscious thinking happens. This cortex is only about 3 millimeters thick, roughly the width of 2 pennies stacked together, yet it contains between 10 and 20 billion neurons, all on its own. The cortex is folded into ridges and valleys called gyri and sulci, allowing a large surface area to fit inside your skull. If you could unfold your cortex and lay it flat, it would be about the size of a large pizza, measuring roughly 1/2 meter on each side. These folds are not random. They follow patterns that allow different regions to communicate efficiently, bringing together areas that need to work as a team. Deep inside your brain, tucked beneath the cortex in a structure shaped like a tiny seahorse, lies the hippocampus. This region is about 5 centimeters long and plays a central role in memory and learning. The hippocampus gets its name from ancient Greek words meaning seahorse, because early anatomists thought it looked like one of those delicate ocean creatures. You actually have 2 hippocampi, one on each side of your brain, nested deep within your temporal lobes near your temples. These structures are constantly busy, processing new experiences and deciding which memories to keep and which to let fade away. When you learn someone's name, navigate through your neighborhood, or remember where you left your keys, your hippocampus is at work, organizing information and linking it to what you already know. The hippocampus does not store memories permanently. Instead, it acts like a librarian, organizing new information and then gradually transferring it to other parts of your brain for long-term storage. This process is called memory consolidation, and it happens mostly while you sleep. During the night, your hippocampus replays the events of your day in fast forward, strengthening the connections between neurons in other brain regions. Over time, these memories become independent of the hippocampus, settling into the folds of your cortex where they can last a lifetime. This is why sleep is so important for learning. Without it, the hippocampus cannot properly file away your new experiences, and new memories remain fragile and easily forgotten. Your hippocampus is also essential for spatial memory, which is your ability to navigate through the world. Scientists have discovered special neurons in the hippocampus that fire only when you are in specific locations. These cells are called place cells, and they create a kind of mental map of your environment. When you walk through your home, different place cells activate depending on which room you are in. Over time, these cells build a detailed representation of your surroundings, allowing you to find your way even in the dark. Some people have larger hippocampi than others, particularly those who spend a lot of time navigating complex environments. London taxi drivers, for example, have been shown to have enlarged hippocampi from years of memorizing the city's winding streets. The hippocampus continues to grow new neurons throughout your life, a process called adult neurogenesis. For many years, scientists believed that you were born with all the neurons you would ever have, and that none could be replaced once lost. But research in the late 20th century revealed that certain parts of the brain, particularly the hippocampus, can generate new neurons even in adulthood. These newborn neurons migrate into the existing network and begin forming connections, potentially helping you learn new information and form new memories. Physical exercise, mental stimulation, and living in enriching environments can all boost the production of these new neurons, suggesting that your brain remains flexible and capable of growth no matter how old you are. Memory is not stored in a single place. It is distributed across your brain in patterns of synaptic connections. When you remember something, you are actually recreating a pattern of neural activity that occurred when you first experienced that event. This means that memories are not like photographs sitting in a file cabinet. They are more like songs that your brain learns to play again, reconstructing the experience from scattered notes stored in different regions. Each time you recall a memory, you are rebuilding it from these fragments, and in the process, you might change it slightly. This is why memories can shift over time, becoming more vivid in some details and fading in others. Your memory is not a recording. It is a performance, recreated fresh each time you remember. Different types of memory involve different brain regions. When you remember facts, like the capital of France or your phone number, you are using declarative memory, which relies heavily on your hippocampus and the surrounding areas. But when you remember how to ride a bicycle or tie your shoes, you are using procedural memory, which involves structures deep in your brain like the basal ganglia and cerebellum. These motor memories are more automatic and less conscious, which is why you can perform them without thinking. Once learned, procedural memories are remarkably stable and resistant to forgetting. You might not have ridden a bicycle in years, but your brain remembers the movements as if you had practiced yesterday. Your brain is divided into 4 main lobes, each responsible for different functions. The frontal lobe sits at the front of your brain, just behind your forehead. It is the largest lobe and takes up nearly a third of your brain's surface. This region is responsible for higher-level thinking, including planning, decision-making, problem-solving, and controlling your behavior. The very front part, called the prefrontal cortex, is involved in personality, judgment, and social appropriateness. It helps you think about the future, weigh consequences, and inhibit impulses that might get you into trouble. Damage to the frontal lobe can change someone's personality dramatically, making them impulsive, disinhibited, or unable to plan ahead. The frontal lobe also contains the motor cortex, a strip of tissue that controls voluntary movement. This area is organized like a map of your body, with different regions responsible for moving different body parts. The amount of space devoted to each body part is not proportional to its size, but to how much fine control it requires. Your hands and face, which need precise movements, take up far more space in the motor cortex than your legs or torso. This arrangement is called the motor homunculus, and if you drew a person based on how much brain space each body part occupies, you would end up with a strange figure with enormous hands and lips and tiny legs. The motor cortex sends signals down your spinal cord to muscles throughout your body, turning intentions into actions. Just behind the frontal lobe, separated by a deep groove called the central sulcus, lies the parietal lobe. This region is involved in processing sensory information from your body and understanding spatial relationships. The parietal lobe contains the somatosensory cortex, which receives information about touch, temperature, pressure, and pain from all over your body. Like the motor cortex, the somatosensory cortex is organized as a map, with neighboring body parts represented in neighboring regions of the brain. When you touch something hot, signals travel from receptors in your skin up to the somatosensory cortex, where they are interpreted as the sensation of heat. The parietal lobe also helps you understand where your body is in space and how to interact with objects around you. The parietal lobe plays a crucial role in spatial awareness. It helps you navigate through your environment, judge distances, and manipulate objects. When you reach for a cup of tea, your parietal lobe calculates the distance between your hand and the cup, the trajectory your arm should follow, and the angle at which to grasp the handle. All of this happens automatically, without conscious effort. The parietal lobe integrates information from your eyes, your muscles, and your sense of touch, create a coherent understanding of where things are in relation to your body. Damage to this area can result in strange conditions like neglect, where a person becomes unaware of one side of their body or the space around them. The temporal lobe sits on the side of your brain, near your temples. This region is involved in processing sound, understanding language, and forming memories. The primary auditory cortex, located in the temporal lobe, receives information from your ears and interprets it as sound. Different areas of the auditory cortex respond to different frequencies, allowing you to distinguish between high notes and low notes, between a whisper and a shout. The temporal lobe also contains regions that help you recognize complex auditory patterns, like speech and music. When you listen to someone talking, your temporal lobe is working to identify the individual sounds, combine them into words, and extract meaning from the sequence. An important area within the temporal lobe is Wernicke's area, which is essential for understanding language. While Broca's area in the frontal lobe helps you produce speech, Wernicke's area helps you comprehend it. Damage to Wernicke's area results in a condition where people can still speak fluently, but their words make no sense, and they cannot understand what others are saying to them. They might produce long, grammatically correct sentences that are completely meaningless, filled with nonsense words and tangled ideas. This shows that language is not a single function, but a network of processes spread across multiple brain regions, each contributing a different piece to the complex task of communication. The temporal lobe also houses structures important for memory, including the hippocampus. The medial temporal lobe, which includes the hippocampus and surrounding areas, is essential for forming new memories. One of the most famous cases in neuroscience involved a patient known as HM, who had his medial temporal lobes removed to treat severe epilepsy. After the surgery, he could no longer form new, long-term memories. He could remember events from before the operation, but everything that happened afterward faded within minutes. He lived in a perpetual present, unable to remember people he met or conversations he had. His case taught scientists that the temporal lobe is critical for moving information from short-term memory into long-term storage. At the very back of your brain lies the occipital lobe, the smallest of the four main lobes. This region is dedicated almost entirely to vision. The primary visual cortex, also called V1, receives signals from your eyes and begins the process of interpreting visual information. When light enters your eyes and strikes your retinas, it is converted into electrical signals that travel along the optic nerve to the back of your brain. The occipital lobe processes these signals, identifying edges, colors, and movements. From there, information flows to other visual areas that recognize shapes, faces, and objects. Your brain constructs the experience of seeing from millions of tiny fragments of information, assembling them into a coherent picture of the world. The visual cortex is organized retinotopically, meaning that the spatial arrangement of the image on your retina is preserved in the arrangement of neurons in your occipital lobe. Neighboring points in space activate neighboring neurons in the brain. This creates a kind of map of your visual field, stretched and distorted, but still maintaining the relationships between objects. Different parts of the visual cortex specialize in different aspects of vision. Some areas respond specifically to motion, others to color, and still others to orientation. Together, these specialized regions work in concert to give you the rich, detailed experience of sight, even though what you perceive feels like a single, unified scene. Your brain consumes a disproportionate amount of your body's energy. Though it makes up only about 2% of your body weight, it uses roughly 20% of your total energy. This energy is needed to maintain the electrical gradients across neuronal membranes, power the pumps that move ions in and out of cells, and fuel the constant communication between neurons. Even when you are resting, your brain remains highly active, using almost as much energy as when you are engaged in a demanding mental task. This background activity is sometimes called the default mode network, a pattern of brain activity that occurs when you are not focused on the outside world, but are instead daydreaming, remembering, or thinking about yourself. The default mode network includes several brain regions that are more active when you are at rest than when you are focused on a task. These areas include parts of the medial prefrontal cortex, the posterior cingulate cortex, and regions of the parietal lobe. When you let your mind wander, these areas light up, suggesting that they are involved in internal mentation, self-reflection, and imagining future scenarios. The default mode network is thought to play a role in constructing your sense of self, integrating memories, and simulating possible futures. It is the brain's resting state, but rest does not mean inactivity. Your brain is always working, even when you think you are doing nothing. Your brain communicates through both electrical and chemical signals. Within a neuron, information travels as an electrical impulse called an action potential. This impulse moves down the neuron's axon like a wave, driven by the flow of ions across the cell membrane. The action potential is an all-or-nothing event. Either it happens or it does not. There are no partial action potentials. This binary nature makes neural communication reliable and fast, allowing signals to travel at speeds up to 100 meters per second in some neurons. The speed depends on the thickness of the axon and whether it is coated in myelin, a fatty substance that insulates the axon and speeds up signal transmission. When the electrical signal reaches the end of the axon, it triggers the release of neurotransmitters, which are chemical messengers. These molecules diffuse across the synaptic gap and bind to receptors on the receiving neuron. Depending on the type of neurotransmitter and receptor, the signal can either excite the receiving neuron, making it more likely to fire its own action potential, or inhibit it, making it less likely to fire. This balance between excitation and inhibition is crucial for normal brain function. Too much excitation can lead to seizures, while too much inhibition can shut down neural activity. Your brain constantly adjusts the strength of synaptic connections, fine-tuning the balance to keep everything running smoothly. There are many different types of neurotransmitters, each with its own role. Glutamate is the main excitatory neurotransmitter in the brain, responsible for most of the fast communication between neurons. GABA is the main inhibitory neurotransmitter, calming neural activity and preventing runaway excitation. Dopamine is involved in reward, motivation, and movement, while serotonin affects mood, sleep, and appetite. Acetylcholine plays a role in attention, learning, and muscle control. These molecules are released and recycled constantly, their levels rising and falling in response to your experiences and internal states. The balance of neurotransmitters influences how you feel, how you think, and how you perceive the world. Your brain is incredibly plastic, meaning it can change its structure and function in response to experience. This property is called neuroplasticity, and it is what allows you to learn new skills, adapt to new environments, and recover from injury. When you practice a new activity, whether it is playing the piano, learning a language, or solving puzzles, the synapses involved in that activity strengthen. The more you practice, the stronger those connections become, and the easier the task feels. This is the basis of learning. Your brain physically reshapes itself based on what you do, carving pathways through repeated use, like water flowing over stone. Neuroplasticity is most pronounced during childhood, when the brain is still developing and forming new connections at an astonishing rate. A young child's brain contains far more synapses than an adult's, a state called synaptic exuberance. As the child grows, unused connections are pruned away, while frequently used connections are strengthened. This process, called synaptic pruning, refines the brain's network, making it more efficient. However, neuroplasticity does not stop in childhood. Adults retain a significant capacity for change throughout life. Learning something new, even late in life, can lead to measurable changes in brain structure, including increased grey matter density in regions involved in the new skill. Different forms of neuroplasticity occur at different timescales. Short-term plasticity can happen in minutes or hours, as synapses temporarily change their strength in response to activity. Long-term plasticity, such as long-term potentiation, can last for days, weeks, or even a lifetime. Long-term potentiation occurs when repeated stimulation of a synapse makes it more responsive to future signals. This is thought to be one of the cellular mechanisms underlying memory. The phrase often used to describe this process is "neurons that fire together, wire together." When two neurons are repeatedly active at the same time, the connection between them strengthens, making it more likely they will activate together in the future. Even the physical structure of your brain can change with experience. Studies of London taxi drivers, who spend years memorizing complex routes through the city, have shown that they have larger hippocampi compared to people who do not navigate for a living. Musicians who practice for thousands of hours show thickening in areas of the motor cortex that control finger movements. People who learn to juggle show structural changes in regions involved in visual motion processing. These findings demonstrate that your brain is not fixed. It is a dynamic organ that continually molds itself based on how you use it, adapting to the demands you place on it and optimizing its structure to match your lifestyle. Your brain undergoes dramatic changes during sleep. Sleep is not a passive state where your brain simply shuts down. It is an active process during which your brain performs essential maintenance, consolidates memories, and clears out waste products. Sleep is divided into several stages, including non-REM sleep and REM sleep, each with distinct patterns of brain activity. During non-REM sleep, which makes up about 75 to 80% of your total sleep time, your brain waves slow down and your body enters a state of deep rest. This is when your brain focuses on physical repair, releasing growth hormones, strengthening your immune system, and consolidating certain types of memories. Non-REM sleep has 3 stages. Stage 1 is light sleep, a transitional state between wakefulness and deeper sleep. Stage 2 is slightly deeper, characterized by bursts of rapid brain activity called sleep spindles. These spindles are thought to play a role in memory consolidation, particularly for motor skills and procedural learning. Stage 3 is the deepest stage of non-REM sleep, sometimes called slow-wave sleep, because of the large, slow brain waves that occur during this period. It is during stage 3 that your body performs most of its physical restoration. If you are awakened from stage 3 sleep, you will feel groggy and disoriented, a state called sleep inertia that can last for half an hour or more. REM sleep, which stands for rapid eye movement sleep, is the stage where most vivid dreams occur. During REM sleep, your brain becomes almost as active as it is when you are awake, but your body is largely paralyzed, preventing you from acting out your dreams. Your eyes move rapidly behind your closed eyelids, and your breathing and heart rate become irregular. The brain regions active during REM sleep include areas involved in emotion, memory, and visual processing, which may explain why dreams are often emotional, illogical, and filled with vivid imagery. REM sleep is thought to play a role in emotional regulation, helping you process difficult experiences and consolidate emotional memories. You cycle through the stages of sleep multiple times each night, with each cycle lasting about 90 minutes. As the night progresses, you spend less time in deep non-REM sleep and more time in REM sleep. Your first REM period might last only 5 to 10 minutes, while your final REM period before waking can last up to an hour. This pattern suggests that different stages of sleep serve different purposes, and your brain needs both to function properly. Deprivation of either non-REM or REM sleep can impair cognitive function, mood, and physical health, highlighting the importance of getting enough high-quality sleep every night. During sleep, your brain also clears out waste products that accumulate during waking hours. A system called the glymphatic system flushes cerebrospinal fluid through the brain, carrying away metabolic byproducts and toxins. This process is most active during sleep, particularly during deep non-REM sleep, when the space between cells expands and allows fluid to flow more freely. One of the substances cleared during sleep is beta amyloid, a protein that can build up and form plaques associated with diseases like Alzheimer's. Chronic sleep deprivation may impair this clearance process, potentially increasing the risk of neurodegenerative diseases over time. Your brain is constantly making predictions about the world. Perception is not a passive process where your brain simply receives information from your senses. Instead, your brain actively generates expectations about what it will experience and then checks those expectations against incoming sensory data. When the prediction matches reality, you perceive the world smoothly and efficiently. When there is a mismatch, your brain updates its model, and you notice something new or unexpected. This predictive coding framework suggests that much of what you perceive is actually a controlled hallucination, a simulation generated by your brain based on past experience and refined by sensory input. This is why context matters so much in perception. The same sensory input can be interpreted differently depending on what you expect to see or hear. Your brain fills in missing information based on patterns it has learned, creating a seamless experience even when the sensory data is incomplete. This is also why optical illusions work. They exploit the assumptions your brain makes about the world, presenting information that contradicts those assumptions and revealing the predictive nature of perception. Your brain is not a camera recording reality. It is an interpreter, constructing a version of the world that is useful for navigating your environment, even if it is not always perfectly accurate. Your brain contains specialized regions for recognizing faces. Faces are so important for social interaction that evolution has dedicated specific areas of the brain to processing them. The fusiform face area, located in the temporal lobe, responds selectively to faces compared to other objects. People with damage to this area can develop a condition called prosopagnosia, or face blindness, where they lose the ability to recognize faces, even those of close friends and family members. They can still see faces perfectly well, but the faces do not trigger the sense of recognition. This shows that face perception is a distinct process, separate from general object recognition. Humans are remarkably good at reading subtle emotional cues from faces. Even newborns show a preference for looking at face-like patterns, suggesting that face recognition is an innate ability. As you grow and gain experience, your brain becomes expert at detecting tiny changes in facial expressions, extracting information about emotions, intentions, and trustworthiness. You can distinguish thousands of different faces and remember them for years, even after a brief encounter. This ability requires not just the fusiform face area, but also a network of regions involved in emotion processing, memory, and social cognition, all working together to help you navigate the social world. Your brain has two hemispheres, a left and a right, connected by a thick bundle of nerve fibers called the corpus callosum. For the most part, the two hemispheres look similar and perform similar functions, but there are some important differences. In most people, language functions are lateralized to the left hemisphere, meaning that areas like Broca's area and Wernicke's area are located in the left side of the brain. Damage to the left hemisphere is more likely to result in language impairments than damage to the right hemisphere. However, this lateralization is not absolute, and the right hemisphere contributes to language processing, particularly for understanding tone, context, and humor. The right hemisphere is often associated with spatial processing, attention, and recognizing patterns. It is better at tasks that require holistic, integrative thinking, like recognizing faces or appreciating music. The left hemisphere, in contrast, tends to be more specialized for analytical, sequential processing, like understanding grammar or performing mathematical calculations. These generalizations are useful, but oversimplified. In reality, most complex tasks require both hemispheres working together. The corpus callosum allows the two sides to communicate and coordinate, ensuring that information is shared and integrated. Cutting the corpus callosum, as was sometimes done to treat severe epilepsy, results in a split brain condition where the two hemispheres operate somewhat independently. Your brain is shaped by both genetics and experience. The basic architecture of your brain is laid out by genes, which guide the formation of brain regions, the migration of neurons, and the initial pattern of connections. However, experience plays a crucial role in fine-tuning this structure. Early childhood is a sensitive period when the brain is particularly responsive to environmental input. Experiences during this time, both positive and negative, can have lasting effects on brain development. Children raised in enriched environments with lots of stimulation, social interaction, and learning opportunities tend to have more complex neural networks and better cognitive outcomes. Conversely, early adversity, neglect, or trauma can disrupt normal development and have long-term consequences. The developing brain is more flexible than the adult brain, but this flexibility comes at a cost. During critical periods of development, the brain is especially sensitive to certain types of input. For example, there is a critical period for language acquisition, during which children can learn languages effortlessly. After this period closes, learning a new language becomes much harder. Similarly, there are critical periods for visual development, social skills, and emotional regulation. If the right experiences do not occur during these windows, it can be difficult or impossible to develop those abilities later. This underscores the importance of providing children with rich, nurturing environments during these formative years. Your brain is constantly balancing opposing forces: excitation and inhibition, activity and rest, growth and pruning. This balance is essential for healthy brain function. Too much excitation can lead to seizures or anxiety, while too much inhibition can result in lethargy or depression. The brain regulates itself through homeostatic mechanisms, adjusting the strength of synapses, the number of receptors, and the release of neurotransmitters to maintain stability. When you repeatedly activate a neural pathway, the brain may down-regulate that pathway to prevent overstimulation. Conversely, if a pathway is underused, the brain may upregulate it to maintain sensitivity. This ongoing adjustment allows your brain to remain responsive to new information while avoiding overload. Your emotions are not separate from your cognition. They are deeply intertwined with your thoughts, memories, and perceptions. The limbic system, a collection of structures deep within the brain, plays a central role in emotional processing. Key components of the limbic system include the amygdala, which is involved in fear and threat detection, and the hippocampus, which links emotions to memories. When you experience something emotionally significant, your amygdala activates, signaling to your hippocampus that this event is important and should be remembered. This is why emotional memories are often more vivid and long-lasting than neutral memories. The intensity of the emotion acts like a highlighter, marking the memory for preferential storage. The amygdala is a small, almond-shaped structure located deep within each temporal lobe. You have two amygdalae, one on each side of your brain, and they are constantly monitoring your environment for potential threats. When the amygdala detects something that might be dangerous, it triggers a rapid response before you are even consciously aware of the threat. This is why you might jump at a sudden loud noise or pull your hand away from something hot before you realize what happened. The amygdala operates on a better safe than sorry principle, erring on the side of caution to keep you safe. This ancient system has been preserved through evolution because it helped our ancestors survive in environments where quick reactions meant the difference between life and loss. The amygdala does not only process fear, it is involved in processing all emotionally salient information, whether positive or negative. When you see something that makes you happy, angry, disgusted, or surprised, your amygdala responds. It helps you recognize emotional expressions in other people's faces, allowing you to quickly assess their emotional state and respond appropriately. The amygdala is also involved in emotional learning, helping you form associations between stimuli and their emotional consequences. If you once ate spoiled food and became sick, your amygdala helps you remember that experience and avoid similar foods in the future. The prefrontal cortex, located at the very front of your brain, helps regulate emotions. It can dampen the response of the amygdala, allowing you to control your reactions and think rationally, even in emotional situations. This ability to regulate emotions develops gradually throughout childhood and adolescence as the prefrontal cortex matures. This is one reason why children and teenagers often struggle with impulse control and emotional regulation compared to adults. When the prefrontal cortex is damaged or underdeveloped, people may have difficulty controlling their emotions, leading to impulsive behavior and mood swings. The interplay between the amygdala and the prefrontal cortex is crucial for emotional balance. When you feel a strong emotion rising, your prefrontal cortex can step in and help you evaluate the situation more rationally. It asks questions like, is this threat really as dangerous as it seems? Or, will this decision that feels good now have negative consequences later? This cognitive control over emotion is what allows you to delay gratification, resist temptations, and make decisions based on long-term goals rather than immediate feelings. It is a uniquely developed capacity in humans, though other primates show some degree of emotional regulation as well. Your brain produces a variety of chemical messengers that profoundly influence your mood, energy, and behavior. These molecules, called neurotransmitters and hormones, circulate through your brain and body, adjusting your internal state to match your circumstances. Serotonin, often called the mood neurotransmitter, plays a role in regulating happiness, anxiety, and overall emotional well-being. Low levels of serotonin have been linked to feelings of sadness and worry, though the relationship is complex and not fully understood. Serotonin is produced in several areas of the brain, particularly in the raphe nuclei of the brainstem, and its effects spread throughout the brain and body. Dopamine is another crucial neurotransmitter, deeply involved in motivation, reward, and pleasure. When you accomplish something meaningful, eat something delicious, or experience something enjoyable, your brain releases dopamine. This release creates a sense of satisfaction and reinforces the behavior that led to the reward, making you more likely to repeat it in the future. Dopamine is essential for learning what actions lead to positive outcomes. However, dopamine is not just about pleasure. It is also involved in motivation and effort. The anticipation of a reward can trigger dopamine release even before you receive the reward, driving you to work toward your goals. The dopamine system can be hijacked by substances and behaviors that provide intense, immediate rewards. Certain substances cause massive releases of dopamine, far beyond what natural rewards produce. This flood of dopamine can overwhelm the brain's reward system, leading to powerful cravings and the pattern of behavior associated with various dependencies. Over time, the brain adapts to these excessive dopamine levels by reducing the number of dopamine receptors, making natural rewards feel less satisfying and creating a need for more of the substance to feel normal. Understanding the dopamine system helps explain why some behaviors can become so compelling and difficult to change. Endorphins are another class of molecules produced by your brain, particularly during physical exertion, stress, or situations involving discomfort. These natural substances act like the body's own comfort agents, binding to receptors in the brain and reducing the perception of unease. This is why intense physical activity can sometimes produce a sense of well-being or calm, often called a runner's calm. Endorphins also play a role in the placebo effect, where believing that a treatment will help can actually reduce discomfort, partly because the belief triggers endorphin release. Your brain has its own internal system for managing difficult sensations, and endorphins are a key part of that system. Oxytocin, sometimes called the bonding hormone, is released during social interactions, particularly those involving trust, intimacy, and connection. When you hug someone you care about, spend time with close friends, care for a child, your brain releases oxytocin. This molecule enhances feelings of attachment and trust, strengthening social bonds. It plays a crucial role in parental care, helping parents feel connected to their offspring. Oxytocin is also released during childbirth and nursing, promoting the bond between parent and infant. In romantic relationships, oxytocin contributes to feelings of closeness and commitment. This chemical system has evolved to support the social connections that are essential for human survival and well-being. Cortisol is a hormone released in response to pressure or challenge. When you encounter a stressful situation, your hypothalamus activates a cascade of signals that ultimately leads to cortisol release from your adrenal glands. Cortisol mobilizes energy, increases alertness, and prepares your body to deal with the challenge. In the short term, this response is adaptive and helpful. However, chronic activation of the cortisol system can have negative effects. Prolonged high levels of cortisol can impair memory formation, weaken the immune system, and contribute to various health issues. The hippocampus is particularly sensitive to cortisol, and chronic tension can actually cause the hippocampus to shrink, affecting memory and learning. Your brain's chemical balance is delicate and influenced by many factors including sleep, diet, exercise, social interactions, and environmental conditions. When this balance is disrupted, it can affect mood, cognition, and behavior. Many therapeutic approaches, both medicinal and behavioral, work by helping to restore or optimize this chemical balance. Understanding the role of these molecules helps explain why certain interventions are effective and highlights the intimate connection between the physical chemistry of the brain and the subjective experience of mental states. The cerebellum, located at the back of your brain beneath the occipital lobe, is a remarkable structure involved in coordinating movement and maintaining balance. Although it is much smaller than the cerebrum, the cerebellum contains more neurons than the rest of the brain combined. Estimates suggest it holds between 55 and 70 billion neurons packed into its tightly folded structure. These neurons are organized in a highly regular, repetitive pattern, creating a kind of neural crystal that is optimized for processing timing and coordination information. The cerebellum does not initiate movements, but it refines them, ensuring they are smooth, accurate, and properly timed. When you reach for an object, your motor cortex sends the command to move, but your cerebellum adjusts the movement in real time, correcting errors and compensating for unexpected changes. If the object is farther away than you thought, or if your hand starts to veer off course, your cerebellum makes rapid adjustments to keep the movement on target. This happens automatically, without conscious awareness, allowing you to perform complex motor tasks with apparent ease. Learning a new physical skill, like playing a musical instrument or a sport, involves training your cerebellum to refine the movements until they become automatic and fluid. The cerebellum also plays a role in cognitive functions beyond motor control. Recent research has revealed that it is involved in attention, language, and even emotional regulation. Damage to the cerebellum can result not only in problems with coordination and balance, but also in subtle cognitive and emotional changes. The full extent of the cerebellum's contributions to cognition is still being explored, but it is clear that this structure is more than just a movement controller. It is an integral part of the brain's network, contributing to many aspects of behavior and cognition. Your brain processes sensory information from multiple modalities, constantly integrating signals from your eyes, ears, skin, nose, and taste buds to create a unified perception of the world. This integration happens so seamlessly that you rarely notice it, but it requires sophisticated neural processing. Different sensory modalities are processed in different brain regions, yet they must be combined to create a coherent experience. When you bite into an apple, you simultaneously experience its taste, texture, smell, and the sound of the crunch. Your brain binds these separate sensory streams together, creating the unified experience of eating an apple. This multisensory integration can be demonstrated through various illusions. In the McGurk effect, what you hear is influenced by what you see. If you watch a video of someone making one mouth movement while hearing a different sound, your brain may perceive a third sound that is a compromise between the visual and auditory information. This shows that perception is not just about receiving sensory input, but about integrating information from multiple sources to arrive at the most likely interpretation of reality. Your brain is constantly making these kinds of calculations, weighing evidence from different senses to construct your experience of the world. Smell is one of the most ancient senses, and it has direct connections to emotional and memory centers in the brain. When odor molecules enter your nose, they bind to receptors on olfactory neurons, which send signals directly to the olfactory bulb at the front of your brain. From there, information travels to the olfactory cortex and to structures in the limbic system, including the amygdala and hippocampus. This direct connection explains why smells can trigger vivid memories and strong emotions. A familiar scent can instantly transport you back to a specific time and place, bringing with it all the emotions associated with that memory. No other sense has such direct access to memory and emotion centers. Taste, or gustation, is closely linked to smell. Much of what you perceive as taste is actually smell. When you eat, odor molecules from the food travel up the back of your throat to your nasal cavity, where they stimulate olfactory receptors. This is why food tastes bland when you have a congested nose. Your tongue can detect 5 basic tastes: sweet, salty, sour, bitter, and umami, which is a savory taste associated with certain proteins. Different regions of the tongue are sensitive to all 5 tastes, contrary to the myth of a taste map with specific zones for each taste. Taste receptors send signals to the gustatory cortex in your frontal lobe, which interprets these signals as flavors. Touch is mediated by receptors in your skin that respond to pressure, vibration, temperature, and various types of stimulation. Different types of receptors are specialized for different kinds of touch sensations. Some respond to light touches, others to deep pressure, and still others to vibrations at specific frequencies. This information travels up your spinal cord to the thalamus, which acts as a relay station, and then to the somatosensory cortex in your parietal lobe. The somatosensory cortex contains a detailed map of your body surface, with more space devoted to areas with high sensitivity, like your fingertips and lips, and less space for areas with lower sensitivity, like your back. Your sense of hearing allows you to perceive sounds ranging from the deep rumble of thunder to the high-pitched whistle of a bird. Sound waves enter your ear and cause your eardrum to vibrate. These vibrations are transmitted through 3 tiny bones in your middle ear to the cochlea, a spiral-shaped structure in your inner ear filled with fluid. Inside the cochlea are thousands of hair cells that convert mechanical vibrations into electrical signals. Different hair cells respond to different frequencies, allowing you to distinguish between high and low pitches. These signals travel along the auditory nerve to the brainstem and then to the auditory cortex in your temporal lobe, where they are interpreted as sounds. Your brain can localize sounds in space by comparing the timing and intensity of sounds reaching your 2 ears. If a sound comes from your left, it reaches your left ear slightly before your right ear, and it is slightly louder in the left ear. Your brain uses these tiny differences to calculate the location of the sound source. This ability is crucial for navigating the environment and detecting potential threats. It also allows you to focus your attention on specific sounds in a noisy environment, a phenomenon known as the cocktail party effect. Even in a crowded room with many people talking, you can tune in to one conversation and filter out the rest. Vision dominates human perception, consuming a large portion of the brain's processing power. Light enters your eye through the pupil and is focused by the lens onto the retina, a thin layer of tissue at the back of the eye containing millions of light-sensitive cells called photoreceptors. There are 2 types of photoreceptors: rods, which are sensitive to dim light and do not detect color, and cones, which respond to brighter light and are responsible for color vision. Humans have 3 types of cones, each sensitive to different wavelengths of light, corresponding roughly to red, green, and blue. The combination of signals from these 3 types of cones allows you to perceive the full spectrum of colors. Information from the retina travels along the optic nerve to the brain. The optic nerves from both eyes meet at a structure called the optic chiasm, where some of the fibers cross to the opposite side of the brain. This arrangement ensures that information from the left visual field of both eyes goes to the right hemisphere, and information from the right visual field goes to the left hemisphere. From the optic chiasm, visual information travels to the thalamus and then to the primary visual cortex in the occipital lobe. Here, the brain begins to analyze the image, detecting edges, orientations, and movements. This information is then sent to higher visual areas that recognize objects, faces, and scenes. Your brain does not just passively receive visual information. It actively constructs your visual experience based on expectations, context, and past experience. Your eyes make rapid movements called saccades, several times per second, jumping from one point to another. During these movements, your vision is briefly suppressed, yet you perceive a continuous, stable visual scene. Your brain fills in the gaps, creating a seamless experience of seeing. Similarly, you have a blind spot in each eye where the optic nerve exits the retina. But you are not aware of these blind spots because your brain fills in the missing information based on the surrounding scene. The visual system is remarkably good at recognizing patterns and objects, even under challenging conditions. You can recognize a face in dim light, from different angles, or partially obscured, demonstrating the brain's ability to extract essential features and match them to stored representations. This ability is not perfect, and the brain sometimes makes errors, which is the basis of optical illusions. Illusions reveal the assumptions and shortcuts your brain uses to interpret visual information quickly and efficiently. They are not failures of perception, but windows into the strategies your brain employs to make sense of the visual world. Attention is the process by which your brain selects certain information to process while ignoring the rest. At any moment, your senses are bombarded with far more information than you can consciously process. Attention acts as a filter, allowing relevant information to reach awareness while suppressing irrelevant information. You can voluntarily direct your attention, focusing on a particular object, sound, or thought. But attention can also be captured involuntarily by salient stimuli, like a sudden loud noise or a bright flash of light. The brain mechanisms that control attention involve a network of regions in the frontal and parietal lobes, working together to enhance processing in relevant sensory areas. When you focus your attention on something, the neurons processing that information become more active, while neurons processing other information become less active. This enhancement and suppression allow the brain to allocate its limited processing resources to the most important information. Attention is not all or nothing, but comes in degrees. You can divide your attention among multiple tasks, though this typically reduces performance on each task compared to focusing on just one. The feeling of being overwhelmed often occurs when demands on your attention exceed your capacity to process information, forcing your brain to switch rapidly between tasks rather than processing them smoothly. Consciousness is perhaps the most profound mystery in neuroscience. What is it like to be you? How does the firing of neurons give rise to the subjective experience of seeing red, feeling joy, or sensing the passage of time? These questions touch on what philosophers call the hard problem of consciousness. Scientists can study which brain regions are active during conscious experiences, and they can manipulate brain activity to alter consciousness. But explaining how physical processes produce subjective experience remains elusive. Some researchers believe that consciousness emerges from the integrated activity of large-scale brain networks. Others propose that it depends on specific types of neural computations or on quantum processes within neurons. Despite the mystery, we know that consciousness has a neural basis. Damage to certain brain regions can alter or eliminate specific aspects of conscious experience. Patients with damage to the visual cortex lose the ability to consciously see, even though their eyes still function. Patients with damage to the parietal lobe may lose awareness of one side of their body or their environment. Anesthesia can reversibly eliminate consciousness by disrupting communication between brain regions. These observations show that consciousness depends on the coordinated activity of distributed brain networks. Consciousness is not located in a single spot, but arises from the dynamic interaction of many regions working together. There are different levels and states of consciousness. During waking, you are fully alert and aware of your surroundings. During sleep, consciousness changes, becoming less focused on the external world and more involved with internal processes. Dreams are a form of consciousness that occurs primarily during REM sleep, characterized by vivid imagery, emotion, and a lack of logical coherence. Some people experience altered states of consciousness through meditation, where awareness becomes highly focused and the sense of self may change. Understanding these different states helps researchers map the neural correlates of consciousness and explore its boundaries. Brain maintains a sense of continuity over time, creating the feeling that you are the same person from moment to moment and year to year. This continuity depends partly on memory, which links your past experiences to your present self. But it also depends on ongoing neural processes that maintain stable patterns of activity. Even as individual neurons fire and fall silent, even as synapses strengthen and weaken, the overall network maintains certain regularities that constitute your identity. This is similar to how a river remains the same river, even though the water flowing through it is constantly changing. You are a pattern, a dynamic process maintained by the coordinated activity of billions of neurons. The brain contains specialized regions for processing different types of information, but these regions do not work in isolation. They are connected by bundles of axons called white matter tracts, which allow different regions to communicate and coordinate their activity. The corpus callosum is the largest of these tracts, connecting the left and right hemispheres. Other major tracts connect the frontal lobe to the parietal lobe, the visual cortex to regions involved in object recognition, and the hippocampus to cortical areas involved in memory storage. These connections form a complex network where information flows in multiple directions, allowing for flexible, context-dependent processing. The brain's network architecture exhibits properties that make it both efficient and resilient. It has small world properties, meaning that any neuron can reach any other neuron through a relatively small number of connections. This allows for rapid communication across the brain. The network also has hub regions that are highly connected and play a central role in integrating information from multiple sources. These hubs are critical for brain function, and damage to them can have widespread effects. The brain's network is also somewhat redundant, meaning that if one pathway is damaged, others can sometimes compensate, allowing the brain to recover at least some function after injury. Your sense of self, your identity, emerges from the activity of your brain. There is no single self-center in the brain. Instead, your sense of self arises from the coordinated activity of many regions, including the prefrontal cortex, the posterior cingulate cortex, and the medial temporal lobe. These areas are involved in self-reflection, autobiographical memory, and thinking about your own thoughts and feelings, a process called metacognition. When you think about yourself, about your past, or about your future, these regions become active. The default mode network, which is active during rest and mind wandering, is thought to play a key role in maintaining your sense of self over time. Consciousness itself remains one of the great mysteries of neuroscience. We know that consciousness arises from brain activity, but we do not yet understand how. How does the firing of neurons give rise to subjective experience, to the feeling of being you, to the redness of red, or the pain of a stubbed toe? This is sometimes called the hard problem of consciousness. Scientists can map which brain regions are active during conscious experiences, and they can study how consciousness changes with brain injuries or anesthesia. But the mechanism that transforms neural activity into felt experience remains elusive. It is possible that consciousness is not a single thing, but a collection of processes, each contributing to the overall experience of awareness. Different states of consciousness involve different patterns of brain activity. When you are awake and alert, your brain shows fast, desynchronized activity, with different regions firing independently. During sleep, especially deep non-REM sleep, your brain waves slow down and become more synchronized, with large groups of neurons firing together. During REM sleep, brain activity resembles waking, but with some key differences, particularly in the prefrontal cortex, which is less active during dreams. This reduced prefrontal activity may explain why dreams often feel illogical, and why you do not question the bizarre events that occur in them. Without the prefrontal cortex fully engaged, you lack the critical thinking and reality testing that you rely on during waking life. Your brain is exquisitely sensitive to oxygen. Neurons require a constant supply of oxygen-rich blood to function. When blood flow to the brain is interrupted, even for a few minutes, neurons begin to die. This is what happens during a stroke, when a clot or rupture blocks blood flow to part of the brain. The affected area becomes starved of oxygen and nutrients, and cells start to fail. The symptoms of a stroke depend on which part of the brain is affected. A stroke in the motor cortex can cause paralysis. A stroke in the language areas can cause aphasia, and a stroke in the visual cortex can cause blindness in part of the visual field. The brain has limited ability to recover from such damage, although neuroplasticity can sometimes allow other regions to take over lost functions. The brain's blood supply is protected by the blood-brain barrier, a selective filter that prevents harmful substances in the bloodstream from entering the brain. The cells lining the blood vessels in the brain are tightly packed, forming a barrier that allows only certain molecules to pass through. Oxygen, glucose, and some nutrients can cross freely, but many toxins, pathogens, and large molecules are kept out. This barrier protects the brain from infections and chemical imbalances, but it also makes it difficult to deliver medications to the brain. Developing drugs that can cross the blood-brain barrier is one of the major challenges in treating neurological diseases. Your brain's activity generates electrical signals that can be measured from the scalp. This measurement is called an electroencephalogram, or EEG. Electrodes placed on your head pick up the summed electrical activity of thousands or millions of neurons firing together. Different patterns of brain waves correspond to different states of consciousness. Alpha waves occur when you are relaxed and awake with your eyes closed. Beta waves occur during active, focused thinking. Theta waves are common during light sleep and drowsiness. Delta waves, the slowest and largest, occur during deep sleep. By analyzing these patterns, researchers can learn about brain function, diagnose disorders like epilepsy, and study sleep. Your brain continues to develop well into your 20s. While most of the basic structure is in place by early childhood, the prefrontal cortex, responsible for executive functions like planning and impulse control, is one of the last regions to fully mature. This process is not complete until the mid to late 20s, which is why adolescents and young adults often take more risks and struggle with decision-making compared to older adults. Myelination, the process of coating axons with insulating myelin, also continues into early adulthood, improving the speed and efficiency of neural communication. This extended period of development allows the brain to remain flexible and responsive to learning and experience during the formative years of life. As you age, your brain undergoes gradual changes. Some cognitive abilities, like processing speed and working memory, tend to decline with age. Neurons may shrink slightly, and the density of synaptic connections may decrease. However, other abilities like vocabulary and crystallized knowledge can continue to improve or remain stable. The aging brain is not uniformly declining. It is reorganizing, trading raw processing power for accumulated wisdom and expertise. Older adults often rely more on both brain hemispheres to perform tasks, a pattern called bilateral recruitment, which may compensate for age-related declines in specific regions. Lifestyle factors can influence how your brain ages. Physical exercise has been shown to promote neuroplasticity, increase the size of the hippocampus, and improve memory. When you exercise, your heart pumps more oxygen-rich fluid to your brain, and your muscles release molecules that support neural health. One such molecule is brain-derived neurotrophic factor, or BDNF, sometimes called fertilizer for the brain. BDNF promotes the growth of new neurons, strengthens existing connections, and protects neurons from damage. Regular physical activity, even moderate walking, can increase BDNF levels and support brain health throughout life. The benefits of exercise on the brain are so significant that some researchers consider it one of the most powerful interventions for maintaining cognitive function as you age. Mental stimulation, such as learning new skills, solving puzzles, or engaging in social activities, can build cognitive reserve, a buffer that helps the brain resist the effects of aging and disease. Cognitive reserve is the idea that some brains can tolerate more damage before showing symptoms, possibly because they have developed more efficient networks or have more connections to draw upon. People who engage in mentally stimulating activities throughout life tend to have higher cognitive reserve, which may protect against cognitive decline and reduce the risk of certain conditions. Learning new skills, especially those that are challenging and require sustained attention, appears to be particularly beneficial. The brain thrives on novelty and challenge, growing stronger when pushed beyond its comfort zone. Diet also plays a role, with certain eating patterns associated with better brain health. Diets rich in antioxidants, which are found in fruits and vegetables, help protect brain cells from oxidative damage caused by free radicals. Omega-3 fatty acids, found in fish, walnuts, and flax seeds, are essential components of cell membranes and support neural communication. Some studies suggest that diets high in processed foods and sugar may have negative effects on brain health, potentially increasing inflammation and impairing cognitive function. While no single food is a miracle cure, the overall pattern of what you eat can influence the health of your brain over time. Sleep is absolutely critical for brain health. During sleep, your brain consolidates memories, clears out toxic waste products, and performs essential maintenance on neural circuits. Chronic sleep deprivation has been linked to impaired memory, reduced attention, mood disturbances, and increased risk of various health issues. The glymphatic system, which clears waste from the brain, operates primarily during sleep, making adequate rest essential for long-term brain health. Most adults need 7 to 9 hours of sleep per night, though individual needs vary. Prioritizing good sleep hygiene, such as maintaining a consistent sleep schedule and creating a restful sleep environment, is one of the best things you can do for your brain. Social connection is another important factor in brain health. Humans are social creatures, and meaningful relationships appear to protect against cognitive decline. Social interaction requires complex cognitive processes, including language, emotion recognition, perspective taking, and memory. Engaging in conversations, maintaining friendships, and participating in group activities all stimulate these processes, keeping the brain active and engaged. Loneliness and social isolation, in contrast, have been associated with increased risk of cognitive decline and various health problems. The quality of social connections matters more than the quantity. Deep, meaningful relationships appear to be more beneficial than numerous superficial ones. Managing pressure and adversity is also important for brain health. while acute challenges can be adaptive, helping you respond to immediate threats. Chronic activation of the pressure response system can have harmful effects on the brain. Prolonged exposure to chemical signals associated with tension can damage the hippocampus, impair memory formation, and alter the structure and function of the prefrontal cortex. Finding healthy ways to manage everyday pressures, such as through mindfulness, physical activity, social support, or professional help, can protect your brain from these negative effects. The brain is resilient, but it needs periods of rest and recovery to function optimally. Your brain is protected by multiple layers of defense. The skull provides a hard outer shell that shields the brain from physical harm. Inside the skull, the brain is cushioned by 3 layers of membranes called the meninges. The outermost layer, the dura mater, is thick and tough. The middle layer, the arachnoid mater, and the innermost layer, the pia mater, are thinner and more delicate. Between the arachnoid and pia mater is a space filled with cerebrospinal fluid, a clear liquid that bathes the brain and spinal cord. This fluid acts as a shock absorber, protecting the brain from impacts and sudden movements. It also helps remove waste products and delivers nutrients to brain tissue. Cerebrospinal fluid is continuously produced by specialized structures in the brain's ventricles, hollow spaces deep within the brain. The fluid flows through these ventricles, around the brain and spinal cord, and is eventually absorbed back into the circulation. The entire volume of cerebrospinal fluid is replaced several times a day, ensuring that the brain's environment remains clean and stable. Problems with the production or drainage of cerebrospinal fluid can lead to conditions where fluid accumulates, putting pressure on the brain and potentially causing damage. The fluid system is yet another example of the brain's sophisticated mechanisms for maintaining its own health and function. The brain's immune system is unique. Unlike other organs, the brain has limited access to the body's main immune cells. Instead, it has specialized immune cells called microglia that constantly patrol the brain tissue, looking for signs of infection, damage, or debris. When microglia detect a problem, they become activated and work to clean up damaged cells, remove unwanted proteins, and support repair processes. Microglia also play a role in refining neural connections during development by pruning away unnecessary synapses. In a healthy brain, microglia maintain a delicate balance between protection and restraint, responding to threats without causing excessive inflammation. When this balance is disrupted, chronic inflammation can contribute to various neurological conditions. Learning is not just about acquiring new information. It is about transforming the physical structure of your brain. Every time you learn something, whether it is a fact, a skill, or a habit, you are changing the connections between neurons. At the beginning of learning, these connections are weak and fragile. You might need to repeat information many times before you can remember it reliably. But with practice, the connections strengthen. Neurons that repeatedly fire together form stronger bonds, making it easier for signals to pass between them. This is the neural basis of learning. What feels like mental effort on the conscious level corresponds to physical changes at the cellular level. Different types of learning engage different brain systems. Declarative learning, which involves consciously learning facts and events, relies heavily on the hippocampus and related structures in the medial temporal lobe. This system allows you to quickly form memories of new experiences and integrate them with existing knowledge. Procedural learning, which involves learning skills and habits, relies more on the basal ganglia and cerebellum. This system operates largely outside of conscious awareness, gradually refining motor programs and automatic behaviors through repetition. Both systems are important, and they often work together. When you learn to drive, for example, you initially rely on declarative knowledge, consciously thinking about each step. With practice, the skill becomes procedural, and you can drive without thinking about it. The spacing effect is a well-documented phenomenon in learning. Information that is studied in multiple sessions, spaced over time, is remembered better than information crammed into a single session, even if the total study time is the same. This effect occurs because spaced practice allows the brain to consolidate memories between sessions, strengthening the neural connections each time. Cramming might feel productive because you can temporarily recall a lot of information, but this knowledge fades quickly. Spaced repetition, in contrast, builds more durable memories that last longer and are more resistant to forgetting. Understanding how your brain learns can help you study more effectively and retain information better. Retrieval practice is another powerful learning strategy. Testing yourself on material, rather than simply rereading it, forces your brain to actively reconstruct the information from memory. This retrieval process strengthens the memory traces, making the information easier to recall in the future. Even when you struggle to retrieve information and make errors, the effort itself can enhance learning, provided you receive feedback to correct the mistakes. Many students intuitively prefer rereading to testing because it feels easier and more fluent. But research consistently shows that retrieval practice leads to better long-term retention. The brain learns best when it is challenged to actively engage with information, not when it passively receives it. Interleaving is yet another effective learning strategy. Instead of practicing one skill or topic until you master it before moving on to the next, interleaving involves mixing different topics or types of problems during practice. This approach feels more difficult and can lead to more errors during practice, but it results in better long-term learning and transfer of skills to new situations. Interleaving works because it forces your brain to discriminate between different types of problems and select the appropriate strategy for each one. This builds more flexible knowledge that can be applied in varied contexts. The brain benefits from challenge and variation, developing stronger and more adaptable neural representations when learning is effortful. Sleep plays a special role in learning and memory consolidation. During waking hours, you encode new experiences, forming initial memory traces in the hippocampus. During sleep, particularly during certain stages of sleep, these memories are replayed and gradually transferred to the cortex for long-term storage. Sleep also helps integrate new information with existing knowledge, finding connections and patterns that were not obvious during waking. This is why you might wake up with a solution to a problem you were struggling with the day before, or why something you studied before sleep is easier to remember afterward. Sleep is not time wasted. It is an essential part of the learning process, when the brain reorganizes and strengthens what it has learned. The phenomenon of forgetting is not simply a failure of memory, but an adaptive feature of how the brain works. If you remembered every single detail of every experience, your memory would become cluttered with irrelevant information, making it harder to retrieve what you actually need. Forgetting allows the brain to discard information that is no longer useful or relevant, keeping the memory system efficient. The forgetting curve describes how newly learned information fades over time unless it is reviewed. The rate of forgetting is steepest shortly after learning and then levels off, meaning that periodic review can dramatically slow the loss of information. Understanding forgetting can help you plan your learning more effectively. Your brain has a negativity bias, meaning it pays more attention to negative information than to positive information. This bias has evolutionary roots. In the environment where humans evolved, missing a positive opportunity, like finding extra food, was less costly than missing a negative threat, like a predator. As a result, the brain evolved to be more sensitive to potential dangers and negative outcomes. The amygdala responds more strongly to negative stimuli, and negative memories tend to be more vivid and persistent than positive ones. This bias can influence your mood and decision-making, sometimes leading to excessive worry or pessimism. Being aware of this bias can help you consciously balance your perspective, actively noticing and savoring positive experiences to counteract the brain's automatic focus on the negative. Your brain constructs your sense of time. Unlike space, which is mapped directly onto sensory receptors, time has no dedicated rote, sensory organ. Instead, the brain infers the passage of time from changes in the environment and from internal processes. Different brain regions contribute to time perception at different scales. The cerebellum and basal ganglia are involved in perceiving short intervals, on the order of milliseconds to seconds, which are important for motor timing and speech. The prefrontal cortex and parietal cortex are involved in estimating longer intervals and maintaining awareness of time over minutes and hours. Your subjective experience of time can vary dramatically depending on context, attention, and emotion. Time seems to pass faster when you are engaged and enjoying yourself, and slower when you are bored or waiting. This is partly because your brain uses the amount of new information processed as a cue for how much time has passed. When you are engaged in a novel, attention-demanding activity, you process a lot of information, but you are so focused that you do not notice time passing. In retrospect, however, the memory of that period seems long because it is rich with details. When you are bored, you notice time passing moment by moment, making it feel slow. But the lack of memorable details makes that period seem short in retrospect. This asymmetry between experienced time and remembered time is one of the curious features of how the brain constructs temporal experience. The brain's ability to imagine future events uses many of the same neural systems as remembering past events. When you envision what might happen tomorrow or plan for next year, your hippocampus and related structures become active, just as they do when you recall yesterday. This suggests that memory is not just about looking backward, but also about looking forward. Your brain uses past experiences to simulate possible futures, allowing you to plan, prepare, and make decisions. This capacity for mental time travel, for projecting yourself into past and future, is one of the most remarkable features of human cognition, and may be unique to our species or shared only with our closest evolutionary relatives. The basal ganglia, a group of structures deep within the brain, play a crucial role in movement, habit formation, and reward processing. These structures include the caudate nucleus, putamen, and globus pallidus, among others. The basal ganglia receive input from many areas of the cortex, process it, and send output back to the cortex via the thalamus, forming loops that influence behavior. When you perform a voluntary movement, the basal ganglia help select and initiate the appropriate motor program. When you learn a new habit, repeated actions strengthen connections within the basal ganglia, gradually making the behavior more automatic and requiring less conscious effort. The basal ganglia are also involved in motivated behavior and learning from rewards. Dopamine neurons in a nearby structure, called the substantia nigra, send projections to the basal ganglia, releasing dopamine in response to rewards or reward-predicting cues. This dopamine signal influences learning, strengthening actions that lead to positive outcomes and weakening actions that do not. Disruption of dopamine signaling in the basal ganglia is involved in certain movement conditions and is also implicated in various aspects of motivation and decision-making. The basal ganglia demonstrate how deeply intertwined movement, motivation, and learning are in the brain's organization. The thalamus sits at the center of your brain, acting as a relay station for sensory information traveling to the cortex. Almost all sensory input, except smell, passes through the thalamus before reaching cortical processing areas. The thalamus is not just a passive relay, but an active processor that filters and modulates sensory signals based on attention, arousal, and internal state. It also plays a role in regulating consciousness and sleep. Damage to the thalamus can result in sensory deficits, changes in consciousness, and disruptions to sleep-wake cycles. This central structure is essential for coordinating the flow of information throughout the brain. The hypothalamus, though small, exerts enormous influence over your body's internal state. It regulates hunger, thirst, body temperature, circadian rhythms, and the release of hormones from the pituitary gland. The hypothalamus monitors signals from your body and your brain, adjusting physiological processes to maintain balance. When you are hungry, neurons in the hypothalamus detect low energy levels and trigger the sensation of hunger and behaviors aimed at finding food. When you are too warm, the hypothalamus initiates sweating and increased circulation to cool you down. This tiny structure is the control center for homeostasis, the process of maintaining stable internal conditions despite changes in the external environment. The brainstem, located at the base of the brain, where it connects to the spinal cord, controls many vital automatic functions. The medulla regulates breathing, heart rate, and circulation. The pons is involved in sleep, arousal, and relaying signals between different parts of the brain. The midbrain contains structures involved in eye movements, auditory processing, and motor control. Damage to the brainstem can be life-threatening because it disrupts these essential functions. The brainstem is evolutionarily ancient, present in some form in all vertebrates, reflecting its critical role in basic survival functions. Your brain maintains awareness of your body's internal state through a process called interoception. Specialized receptors throughout your body monitor conditions like heart rate, respiration, gut activity, and the state of your organs. This information travels to the brain, particularly to the insula, a region tucked deep within the lateral fissure. The insula integrates these internal signals with emotional and cognitive processing, creating your subjective sense of bodily feelings. This is why emotions are often accompanied by physical sensations, like a racing heart when anxious or a churning stomach when nervous. Your emotional experiences are deeply rooted in your body's physiological state. The brain has remarkable compensation abilities. When one sense is lost, such as vision or hearing, the brain regions that would normally process that sense can be recruited for other purposes. Blind individuals often show enhanced auditory and tactile abilities, and brain imaging reveals that their visual cortex, rather than remaining idle, becomes active during auditory and tactile tasks. This cross-modal plasticity demonstrates the brain's drive to make use of available neural resources, reorganizing itself to optimize function given the input it receives. The brain is not hardwired with fixed functions, but adapts its organization based on experience. Mirror neurons, discovered in the motor cortex of monkeys, are neurons that fire both when an animal performs an action and when it observes another animal performing the same action. This discovery has fascinated neuroscientists because it suggests a neural basis for understanding others' actions, and perhaps for imitation and empathy. The existence and role of mirror neurons in humans are still being investigated, but the concept has influenced thinking about social cognition, language acquisition, and how we understand the intentions and emotions of others. The ability to internally simulate what others are doing or feeling may be fundamental to social interaction and cooperation. Your brain processes faces differently than other objects. Within a fraction of a second after seeing a face, specialized regions in your brain have already identified it as a face and begun analyzing its features, expression, and identity. This rapid processing reflects the social importance of faces. Throughout human evolution, being able to quickly recognize faces and read expressions has been crucial for social survival. Your brain is so attuned to faces that it sometimes sees them in random patterns, a phenomenon called pareidolia. When you see a face in a cloud, a rock formation, or a piece of toast, your brain's face detection system is working overtime, finding patterns that match its template for faces. Emotions serve important functions, guiding behavior and decision-making. Far from being irrational disruptions to clear thinking, emotions provide valuable information about what matters to you and help prioritize actions. Fear motivates avoidance of danger. Happiness reinforces beneficial behaviors. Sadness signals loss and prompts withdrawal and reflection. Even complex emotions, like guilt, pride, and compassion, serve social functions, helping you navigate relationships and maintain group cohesion. The brain's emotional systems are not separate from, but deeply integrated with cognitive systems, jointly contributing to adaptive behavior. The brain's stress response system involves a cascade of hormones and neural signals that prepare your body to deal with challenges. When you encounter a potential threat, the hypothalamus activates the sympathetic nervous system, triggering the fight or flight response. Your heart rate increases, your breathing quickens, energy is mobilized, and your senses sharpen. At the same time, the hypothalamus signals the pituitary gland to release hormones that ultimately cause the adrenal glands to release cortisol. This stress response is adaptive in the short term, helping you deal with immediate challenges. But chronic activation can have negative health consequences. The brain distinguishes between acute and chronic stress responses. Acute stress responses are brief and followed by recovery, allowing the body to return to baseline. Chronic stress, where the system remains activated over long periods, can lead to wear and tear on multiple bodily systems, including the brain. The hippocampus is particularly vulnerable to chronic stress, and prolonged exposure to stress hormones can impair its function and even cause it to shrink. This may partly explain why chronic stress is associated with memory problems and increased risk of certain mental health conditions. Managing stress through healthy coping strategies is important for protecting brain health. Motivation arises from the interaction of multiple brain systems, including those involved in reward, emotion, and goal-directed behavior. When you set a goal and work toward it, your brain is balancing the anticipated reward of achieving the goal against the effort required and the delay until you receive the reward. The prefrontal cortex evaluates these factors and helps maintain focus on long-term goals, even when immediate rewards might pull you in other directions. The dopamine system signals the value of potential rewards and energizes behavior toward obtaining them. When these systems work well together, you feel motivated and capable of sustained effort. When they are disrupted, motivation can falter. The concept of willpower or self-control involves the prefrontal cortex exerting control over more automatic, impulse-driven behaviors. When you resist eating a tempting dessert to stick to a health goal, your prefrontal cortex is inhibiting the reward system's urge to seek immediate gratification. Self-control appears to be a limited resource that can be depleted with use, a phenomenon sometimes called ego depletion, though the robustness and interpretation of this effect are debated. Regular practice of self-control in one domain may strengthen it in others, suggesting that self-control is like a muscle that can be trained. Understanding the brain basis of self-control can help in developing strategies to improve it. The brain's reward system can be influenced by expectations. The placebo effect is a well-documented phenomenon where believing that a treatment will help can produce real physiological changes and symptom relief, even if the treatment has no active ingredients. Brain imaging studies show that placebos can activate the same pain relief systems as actual medications, releasing endorphins and other natural comfort molecules. This demonstrates the power of expectation and belief to influence brain function and bodily states. The placebo effect is not just mind over matter, but a genuine interaction between cognitive processes and physiological systems. The brain is also capable of the opposite phenomenon, the nocebo effect, where negative expectations lead to worsened symptoms or side effects. If you believe a treatment will cause certain discomforts, you are more likely to experience them, even if the treatment is inert. This highlights how your thoughts and expectations, processed by your brain, can have real consequences for your physical experience. Understanding these effects can help in clinical settings, where how treatments are framed and explained can influence their effectiveness. Your ability to pay attention, to focus on one thing while ignoring others, involves a complex network of brain regions working together. The frontal eye fields and parietal cortex help direct your attention to specific locations in space. The prefrontal cortex maintains focus on goals and suppresses distracting information. The thalamus regulates the flow of sensory information to the cortex, enhancing relevant signals and suppressing irrelevant ones. When these systems work smoothly, you can concentrate deeply on a task. When they are disrupted or overwhelmed, attention becomes scattered and focus becomes difficult. Factors like sleep deprivation, certain conditions, and high stress can all impair attentional systems. Your brain is capable of remarkable feats of creativity and imagination. You can generate new ideas, envision scenarios that have never happened, and create art, music, and literature. Creativity is not confined to a single brain region, but involves a network of areas that work together. The default mode network, which is active during rest and mind wandering, is thought to play a role in creative thinking by allowing the free flow of associations and the generation of novel ideas. The prefrontal cortex helps evaluate and refine these ideas, filtering out the ones that are impractical or irrelevant. The interplay between spontaneous idea generation and deliberate evaluation is what makes creativity possible. Your brain is also capable of empathy, the ability to understand and share the feelings of others. When you see someone in pain, mirror neurons in your brain may activate as if you were experiencing the pain yourself. This neural resonance helps you understand what others are feeling and respond with compassion. Empathy involves multiple brain regions, including areas involved in emotion processing, perspective taking, and social cognition. It is a complex, multifaceted ability that allows you to connect with others, build relationships, and navigate the social world. Language is one of the most complex and uniquely human abilities, and it relies on a distributed network of brain regions. Producing speech involves coordinating dozens of muscles in your mouth, throat, and lungs, all guided by signals from the motor cortex. Understanding speech requires processing the sounds, segmenting them into words, accessing their meanings, and integrating them into the context of the conversation. Reading and writing add additional layers of complexity, involving visual processing, symbol recognition, and the translation between written and spoken forms. Despite this complexity, most people acquire language effortlessly during childhood, suggesting that the brain is specially equipped for this task. Your brain is not a static entity. It is a dynamic, living organ that changes from moment to moment, adapting to your experiences, responding to your environment, and shaping who you are. Every thought you have, every memory you form, every skill you learn leaves a trace in the structure of your brain. Over time, these traces accumulate, creating a unique pattern of connections that is yours alone. No two brains are exactly alike, even in identical twins, because experience shapes the brain in ways that genes cannot fully prescribe. Your brain is the physical manifestation of your life, a record of everything you have done, learned, and felt. Understanding the brain is one of the great challenges of science. It is both the tool we use to investigate the world and the object of that investigation. Studying the brain requires us to use the brain to understand itself, which creates a curious loop. Despite decades of research and tremendous advances in technology, much about the brain remains unknown. We are still uncovering the basic principles of how neurons communicate, how networks of neurons give rise to behavior, and how consciousness emerges from this activity. Each discovery raises new questions, and the more we learn, the more we realize how much we do not yet know. Yet this mystery is part of what makes the brain so captivating. It is a frontier, a wilderness of complexity and wonder that we are only beginning to explore. With every experiment, every scan, every insight, we come a little closer to understanding the organ that makes us human. And as we learn more about the brain, we gain not just scientific knowledge, but also practical tools for improving health, treating disease, enhancing learning, and enriching life. The study of the brain is ultimately the study of ourselves, an attempt to answer the age-old questions. What are we? How do we think? What does it mean to be conscious? Your brain is always working, even when you are not aware of it. While you read these words, your brain is processing visual information, interpreting symbols, accessing meanings, constructing sentences, and integrating the content with what you already know. It is regulating your breathing, monitoring your posture, tracking sounds in the environment. It is anticipating what comes next, generating questions, and forming connections between ideas. All of this happens automatically, beneath the surface of conscious awareness, a testament to the incredible complexity and efficiency of the neural machinery that fills your skull. Your brain is a universe in miniature, a cosmos of connections more vast than the night sky. Within its folds lie the secrets of who you are, the memories of everything you have loved, the blueprints for everything you will become. It is both ancient and new, shaped by millions of years of evolution and by every moment of your life. It is fragile, vulnerable to injury and disease, yet resilient, capable of healing and adaptation. It is the seat of your thoughts, the source of your feelings, the keeper of your stories. It is, in every sense, the home of your mind. The more we learn about the brain, the more we appreciate its elegance. It is not a collection of separate parts, but an integrated whole, with every region connected to every other, communicating in a constant symphony of activity. The boundaries between perception and action, thought and emotion, memory and imagination are blurred, revealing that the brain does not operate in neat compartments, but through the continuous interplay of systems. This interconnectedness is what gives the brain its power, allowing it to be more than the sum of its parts, to generate emergent properties that cannot be predicted from studying individual neurons alone. Your brain is also remarkably efficient. Despite its complexity, it operates on about 12 watts of power, roughly the same as a dim light bulb. It packs 86 billion neurons and 600 trillion synapses into a volume of about 1.4 liters. It can process vast amounts of information in parallel, perform pattern recognition that surpasses the best computers, and adapt to new challenges on the fly. It is both a supercomputer and a biological organ, subject to the same constraints and vulnerabilities as the rest of your body, yet capable of feats that we are still striving to replicate with machines. Science continues to push the boundaries of what we know about the brain. New imaging techniques allow us to watch the brain in action, seeing which regions light up during different tasks. Genetic tools let us manipulate specific neurons, turning them on or off to understand their role in behavior. Computational models simulate neural networks, testing theories about how information is processed. Brain-computer interfaces are beginning to bridge the gap between mind and machine, allowing people to control devices with their thoughts or restore lost senses with electronic implants. Each of these advances brings us closer to unlocking the brain's secrets and harnessing its potential. Yet, for all our progress, the brain retains its mystery. We can describe its parts, map its connections, and measure its activity, but the full picture remains elusive. The leap from neural activity to conscious experience, from synaptic firing to the feeling of seeing a sunset, or tasting chocolate, or falling in love, is still not fully understood. This gap is not just a scientific puzzle, but a philosophical one, touching on the deepest questions about the nature of reality, the relationship between mind and matter, and what it means to be a thinking, feeling being. As you lie here, your brain is quietly at work, orchestrating the intricate dance of neurons that underlies your thoughts and feelings. It is preparing your body for sleep, releasing hormones that make you drowsy, slowing down neural activity in regions associated with alertness, and preparing to enter the restorative cycles of non-REM and REM sleep. As you slip into slumber, your brain will continue its work, consolidating memories, clearing waste, and maintaining the essential functions that keep you alive. Sleep is not an absence of activity, but a different mode of operation, one that is just as important as waking. Your brain is a gift, a 3-pound miracle that gives you the ability to perceive, to think, to remember, to imagine, and to feel. It connects you to the world and to other people. It stores your history and shapes your future. It is the organ that makes you you, the foundation of your identity, the source of your consciousness. Caring for your brain means caring for your whole self, recognizing its needs for rest, nourishment, stimulation, and connection. In doing so, you honor the extraordinary complexity and beauty of the biological machinery that makes your life possible. The story of the brain is a story of connection. Neurons connecting to neurons, regions connecting to regions, the brain connecting body to world and mind to mind. It is through these connections that we learn, grow, and adapt. It is through these connections that we form relationships, build societies, and create meaning. Your brain is not an island. It is part of a larger network that includes other brains, other minds, other lives. When you communicate with someone, your brain is engaging with theirs in a dance of signals and symbols, bridging the gap between two separate minds and creating a shared understanding. There is something deeply poetic about the fact that the universe has given rise to brains capable of understanding themselves. From the simplest atoms forged in the hearts of stars to the complex molecules that make up cells, to the intricate networks of neurons that create minds. The cosmos has evolved to the point where it can look back on itself with wonder. Your brain is a piece of the universe that has become self-aware, a small corner of reality contemplating its own existence. In studying the brain, we are studying not just an organ, but the very nature of thought, consciousness, and being. And so, as you rest tonight, know that your brain is both a miracle and a mystery, a masterpiece of biology and a testament to the power of nature. It has carried you through this day, processing your experiences, guiding your actions, and keeping you safe. Now it will carry you through the night, weaving dreams from the threads of memory, repairing and renewing itself, preparing for another day of wonder and possibility. Let your thoughts slow, let your mind settle. Let the tireless work of your 86 billion neurons fade into the background, into the quiet hum of rest. Thank you for spending this time exploring the hidden universe inside your head. As we have wandered together through the folds of the cortex, the depths of the hippocampus, and the networks that create your thoughts and dreams, we have glimpsed the remarkable machinery that makes you who you are. Every memory you treasure, every skill you have learned, every person you have loved, exists because of the silent work of these tiny cells, communicating in a language of electricity and chemistry that predates words. If you found peace in this journey, I invite you to like, subscribe, or leave a comment below. It helps other seekers find this quiet place, one resting mind at a time. But for now, let it all go. Let the details fade. Let your breathing deepen. Your brain will take care of the rest, as it always has, as it always will. It will keep your heart beating, your lungs filling, your body healing. It will sort through the day, filing away what matters, letting go of what does not. You do not need to do anything now. You have done enough. You have learned enough. You have thought enough. Close your eyes and let yourself sink into the softness of sleep, trusting that your brain will continue its work, unseen and untiring, through the long hours of the night. Rest in the knowledge that you are a conscious being, a thinking creature, a mind looking out at the world from inside a 3-pound universe. The stars outside your window are made of the same atoms as the neurons in your brain. You are not separate from the cosmos. You are a part of it, a small but significant thread in the immense weave of existence. And tonight, as you sleep, you rejoin the rhythm of the universe, the ancient cycle of rest and renewal that all living things share. Let the night hold you. Let sleep come. Let your brain, your miraculous, mysterious brain, carry you into dreams. Good night.